Hello students, welcome to Aakash. I'm your botany faculty Dr. Sukirti, and today we are here to discuss this particular paper of AIATS PLUS 2025 Phase 2 Mock Test 8, which was held on 9th of March 2025. So in this particular paper, we are not only going to deal with the grey areas, the questions that you have committed most mistakes in, but also we are going to cover the entire periphery of that topic as well, right? So in this particular session, what we are going to discuss is certain chapters of botany, like we are going to talk about principles of inheritance and variation, cell cycle and cell division, respiration, ecosystem and microbes, right? So let's quickly discuss those questions. The very first one over here is the given diagram of cell represents which stage of mitosis, right? So जब भी हम mitosis की stages की बात करते हैं, what all we can know about it, that in the karyokinesis of it will begin with the karyokinesis, in which we will be having 4 stages, and they will be beginning with prophase, followed by metaphase, then anaphase, and finally telophase. Right? So when we talk about the prophase, what all we get from here? In prophase, very key characteristic events are, firstly, the chromatin that is being present in the interphasic nucleus, right? That particular chromatin is going to get condensed in the prophase, right? We'll be seeing this sort of condensation, this will start. Apart from that, one more event that we are going to notice is the disintegration of the nuclear membrane, right? Also, we will be seeing the formation of mitotic apparatus over here, and in case of animal cells, we'll also see asters over here. So asters, along with the 2 asters on 2 poles, along with the spindle fiber, is going to form the mitotic apparatus, right? So that formation also going to take place here. The disintegration of nucleolus is also seen, and the various organelles that we see in the cell is now not going to be appeared in this particular cell, which is in the prophase stage. Further, when the prophase is completed, right, how we are going to know that the prophase is completed? So when there is a complete disintegration of the nuclear envelope, that is going to mark the beginning of the next stage. So this is the prophase, and the next stage after the complete disintegration would be metaphase. So in this particular phase, what we are going to observe, that the condensation is completed now. The chromosomal condensation is completed. The nuclear membrane has already been disintegrated, and now the congression will start, right? You may have heard this particular word, the congression, right? So congression is basically means that the chromosomes are now going to come to the equator part, and in this phase we are going to see that the spindle fibers are going to get attached with the kinetochore region, right? Now this particular phase is followed by anaphase, right? And because we are talking about mitosis here, which is an equational division, so the characteristic event of anaphase in mitosis is the splitting of centromere, right? That will cause the segregation of chromatids, and now these chromatids, which will now be referred to as the chromosomes, are going to go towards the opposite poles, right? Then further, this is going to be followed by the last phase of karyokinesis, which is telophase. And in this, what we are going to observe is the decondensation of these chromosomes at the 2 poles, and the reappearance or reintegration of the nuclear envelope, the nucleolus, and so on, right? After this karyokinesis is being completed, that means the division of nucleus is completed, the cell will now perform cytokinesis, right, in which the cytoplasmic division is now going to take place. So whatever organelles are going to be present, or the cytoplasmic content is going to be present, that is going to be equally distributed to the two daughter cells. And as we can clearly see that the ploidy of the mother cell and the daughter cells are still remain same, that is why we call this particular mitosis division as the equational division, right? So, if after this particular karyokinesis is completed and cytokinesis is not occurring, then we could clearly see that in a single cell we will be observing two nuclei. So, if we are going to see this concurrently, that means the karyokinesis is occurring but cytokinesis is not occurring, then we are going to see the formation of syncytium, a multinucleate condition, right? So, this is about the phases of mitosis, right? So, how exactly the questions could be asked? They could ask you about the features of, like what kind of features are going to be exhibited by a particular phase, or they can give you the microscopic figures of it, like we can see in the question also. Over here they have given a microscopic image. So, from this image, how we could decipher that which phase we are talking about. As we have discussed that in the very first phase of the karyokinesis of mitosis, we see the disintegration of the nuclear membrane. If you could see here, you can see there are certain breaks that are being present in this nuclear membrane. This is the cell membrane, right? And over here in the nuclear membrane, you can see these breaks, right, which is basically inferring the fact that it is disintegrating, right, in the very early one. So, and the chromatin, right, that is being present inside it is still not in the form of chromosome, right, it has just started it. So, that means it is most probably inferring towards the early prophase. Correctly written by Tanmay, wonderful. So, this is the early prophase they are depicting. In the late prophase, what we are going to observe is the disintegration is almost being done. Then we will see if we consider it as an animal cell, we will see the formation of aster here. Also, this particular chromatin condensation would almost going to be, these chromosomes are now going to be look like an egg-shaped structure, right, when the late prophase is going to be. So, this could be crossed out, right, it could be ruled out because over here it's still not happening. In metaphase, they could have been aligned on this particular plate, metaphasic plate, that's also not occurring. So, this could also be ruled out. And transition to metaphase is basically the late prophase, right, because we have said this that the complete disintegration of the nuclear membrane is the beginning of the metaphase, right, and that is going to be completed in the late prophase. So, this transition to metaphase is also referring towards the late prophase. So, we could rule out this as well. So, the correct answer for this particular question is going to be option number 4, right, correctly said by too many students. So, wonderful. I hope this particular question is clear to everyone. So, now let us move ahead to another question, and this is talking about all are the features of final stage of mitosis, karyokinesis, except, right. So, as we have discussed the karyokinesis already, right, that in the prophase what all events are going to take place, in the metaphase the congression and the alignment on the equatorial plate, right, and then further in anaphase chromatid splitting, and then in telophase decondensation occurs. So, with that particular information, if we see that what are the features of final stage of mitosis of karyokinesis, they have specifically mentioned this. So, if we see the first option which says chromosome lose their identity as discrete elements, right. So, we could rule out this particular one, right. Why? Because when we say that chromosome lose their identity as discrete elements, that means we are referring towards the decondensation of it. And this question is asking that all of them are the features of the final stage of karyokinesis. Final stage means they are talking about telophase, right, and we have to talk about the one that is not the feature of it, right. So, this is the decondensation, characteristic event of telophase. Then second one, nucleolus Golgi complex and ER reformation. Of course, this also occurs during telophase. Their disintegration will be in the prophase, but their reformation is in the telophase stage. Then third one is saying nuclear envelope reforms, again event of telophase, right. Then fourth is compaction of chromosome. This is an event of prophase where it begins. And by metaphase, this condensation is completed, right. So, this is the only event which does not occur in telophase. That is why the correct answer over here would be option number 4, right? Correctly said by Tanmay, 4 is the answer, as chromosome lose their identity as discrete elements given in option 1, right? Alright, let's move ahead to another question. That's question number 105, which talks about the quiescent stage of cell is, right? So when we talk about the cell cycle, right? So we will be having a very small phase in which the division occurs, but in this entire phase the cell is preparing for it, right? So this is the interphase. Or the resting phase, because we are not going to observe any of the events that occur over here under the microscope. That is the reason why we call it as a resting phase, not because the cell is resting, right? So in this particular phase, when the cell is preparing itself for division, in that case what it does, firstly it will be having this G1 phase, which is the gap 1 phase. We also called it as post-mitotic phase because it is going to occur after M phase, right? The cell begins with it, but of course it is after the M phase is completed, right? It begins with it, so it will be G1 phase. So in this phase, what will happen? Over here, we'll be having synthesis of proteins, right? Other biomolecules as well, and over here, most of the organelles will be duplicating itself. Right? Now, if in case the cell has to differentiate or because of certain other stimuli, the cell exit from here, right, then it will enter into G0 phase, that is also known as quiescent phase, right? So quiescent phase is the phase in which the cell is going to perform its function, right? Plant cell undergoes quiescent stage in cell cycle, and this leads them the property of dedifferentiation and redifferentiation. See, G0 is the phase where the differentiation, is going to occur, because now the cell is being destined to perform certain function. If the cell is undergoing dedifferentiation, then it is getting back into the cell cycle in the G1 phase so that it could perform all the other activities. So if either it is differentiation or redifferentiation, in which the cell is going to exit from the G1, enter into the G0, and till the stimuli is not getting back, till the time it is going to retain in the G0 phase, perform its metabolic activity, but not going to proliferate, right? So this is the quiescent stage. And then if let's suppose this is not being there and the cell has to divide, right? And this will happen if the nucleocytoplasmic ratio is going to get disturbed. In that case, of course, the cell is going to divide itself. In that particular case, after the organelle duplication is being done, right, a major amount of cell growth is also going to be happening in this particular phase. Then the cell will enter into S phase, which is the synthesis phase. The characteristic event of this particular phase is the DNA replication, right? This is a very important and a key event, and the reason for that particular one is because this is the genetic material and it has the entire information that how the cell has to perform its various activities, the metabolic activities. So the control is over here. That is the reason why S phase is so much predominant and clearly specific phase, because over here DNA replication is going to take place, and this is the only phase where DNA replication is occurring, and it's not going to happen anywhere else, right? So after this S phase is done, in this S phase, if it is a animal cell, right, in that particular case, or the cells that are having centriole, in those cells, centriole duplication will also take place here, right? Then further after this, the cell will enter into G2 phase, which is also known as the pre-mitotic phase. Over here also the protein synthesis take place. Specifically, the protein that is going to be there will be tubulin, because this particular, protein is going to be used for the synthesis of spindle fibers, right? And then the cell could enter into M phase to perform karyokinesis and cytokinesis that we have talked about, right? So if the phase is present, if the cell is being present in G1S or G2 phase, it is destined to perform M phase. If all the events are going to be happening, right? Like if let's suppose there are like 10 types of proteins that needs to be made for S phase, if all those are going to be made, only then the cell will be allowed to enter from G1 to S phase. If any of the function is occurring inaccurately, in that particular case, what will happen? The checkpoints of this particular cells will block it, right? So that is how the cell cycle is regulated, but only when, you know, the cell has to perform certain function, perform the differentiation part, then it will exit the G1 phase and enter into G0 phase, correct? Alright, so that's what quiescent stage is all about. So if we are getting back to this, and it says that the quiescent stage of cell is. So the first one is saying an inactive stage in terms of cell division. So yes, this is true, because in the G0 phase, the cell is performing its metabolic functions, but it is not proliferating. It's not like that it will always retain into that, but it could come back into the cell cycle phases only if there is a stimulus for it. So this is inactive in terms of cell division. Then the option 2 is saying an abnormal stage of actively dividing cell. That is incorrect. This particular abnormal division is shown by the cancerous cells, which are like far away from the checkpoints. So they are continuously dividing even if all the functions are not being performed accurately in the phases. Now third says seen only in the meristematic tissues. That is incorrect, because when we talk about the meristematic phase cells, those cells are actively dividing cells, right? And as we have specifically mentioned that the cells that are being there in the quiescent stage are unable to proliferate, right? Now option 4 says they are not found in higher animals. This is incorrect. They could be found, like we can say, see the neurons that are being arrested in this particular phase and won't divide any further, right? So in terms of quiescent stage, the correct answer would be option number 1. I hope this is clear to everyone. In case you have any doubts, please put on the chat box so that we could discuss and clear it here only, right? All right, let's move ahead to another question, which is question number 109, and it says that the site of citric acid cycle in eukaryotes is, right? So when we talk about the respiration in organisms, right? So the most favored one, we have glucose, right? This glucose is going to perform glycolysis, right, which will yield pyruvate. This particular reaction, set of reactions of course, are going to occur in the cytoplasm. And as we know that cytoplasm is present in all types of cells, right? So that is the reason why we are going to see that glycolysis is going to occur in all type of organisms. In which phase would mitochondria, ER, plastid, etc. duplication takes place, G1 or G2? When we talk about the mitochondrial duplication and plastid duplication, right, that particular duplication is going to occur in G2 along with Golgi complex. Rest all will be in G1, right? I hope this is clear to everyone. Okay, getting back to the question of respiration. So when we talk about this particular set of reaction, it's occurring in cytoplasm. Now, depending upon the condition, which type of organism is, the pyruvate could enter either into the aerobic functioning or aerobic reactions or anaerobic reactions. If it chooses to have aerobic pathway, then it will perform the link reaction, also known as the gateway reaction, because this is allowing this pyruvate to get access into the mitochondria, right? So this is going to enter into the mitochondrial matrix. So this is acetyl-CoA that is going to be formed. So this pyruvate, which is present in the cytoplasm, enters into the mitochondrial matrix by this particular reaction, which is catalyzed by pyruvate dehydrogenase complex. Now this acetyl-CoA will enter into the Krebs cycle. And whatever reduced coenzymes would be made, they are going to perform ETS, right? They are going to perform ETS so that ATP can be made from those reduced coenzymes, and again those reduced coenzymes gets back into the oxidized form so that these cycles could occur in their original way, right? Now for their uninterrupted continuation, it's important that they have the oxidized coenzyme. That is why as soon as these coenzymes are being made, they are being fed into the ETS so that they can become oxidized, so that these cycles can be performed uninterruptedly. Also at the same time ATP could be formed. But in case if this pyruvate chooses to go for the anaerobic pathway, then it will perform fermentation, right? So from here to here, everything is occurring in mitochondria, right? Then fermentation again could be of two types. We can have alcoholic fermentation and it could have lactic acid fermentation. Right? It could have lactic acid fermentation as well. So this is how the pyruvate can go into either of the pathways, depending upon what kind of conditions are present. If oxygen is abundant, then it would go to this particular pathway. If it's not, or the organism doesn't survive the oxygenic conditions, then this is the pathway by which it could produce ATP and at the same time can also produce the oxidized coenzyme back so that this could continue, right? So this is what is going to be taking place. So as we know that Krebs cycle, because it's being discovered by Hans Krebs, so that's the name it got, the Krebs cycle. Apart from that, it has two more names. One is citric acid cycle. Why we call it a citric acid cycle? Because the first product, right, the stable product that's been made in this cycle is citric acid. So that's why citric acid cycle. And we can also call it as a tricarboxylic acid cycle because citric acid has got 3 carboxylic group in it, right? So that is why. So there is a question, how can I remember ETC to long term? I just forgot, don't know why. So in order to, learn it, for this particular one, you have to practice it concurrently, right? Make the diagrams because the diagrammatic view is much more clear, right? It's not that difficult if you'll continuously practice it. So as many times you'll practice, you are not able to forget it, right? Okay, so getting back to this one, it is talking about the site of citric acid cycle. As I've told you that Krebs cycle is basically citric acid cycle in other way. So the site of this particular cycle would be the mitochondrial matrix, right? When we talk about mitochondrial membrane, this is the site where ETC is going to take place, not the Krebs cycle. And then we can have oxysome. This is again incorrect. That means we are referring towards F0, F1 particle of the ATP synthase molecule, right? And this F0, F1 particle is going to be embedded into the inner mitochondrial membrane. Now when we talk about cytosol, this is also incorrect. This is the place where we are going to have our glycolytic pathway occurring, right? All right, so this is what, this is how we are going to answer this particular one. Also, respiration has got many reactions, right? But there are certain key points that you have to remember because there are been asked multiple times. So learn them in ways like what are the substrate level phosphorylation reactions. Those are important. In glycolysis, the reactions in which ATP is being consumed and is being released and the yields of it, right? That is very important to understand. So in citric acid cycle, they may ask you other questions in terms of where the substrate level phosphorylation occurs, how many times decarboxylation occurs in the cycle, right? So these key features are important, not exactly the reactants and products, but much more in these terms. You have to analyze the cycle much more. The more you'll analyze, the more you will have the retainance of this particular memory for a long time, right? All right. Now let's move ahead to another question. which is question number 112, and it talks about experimental verification of the chromosomal theory of inheritance was done by, right? So earlier when the Mendelian genetics was into the picture, so Mendel was not clear about the physical identity of the molecule that he was constantly calling as factor, that what factor is all about. He said that there is certain thing which is inherited from one generation to the next. Then only, like in later 1900s, when De Vries, Correns and Teschmack have rediscovered those laws, they have said that this could be gene. But then later on, two people, which are Sutton and Baveri, came up with the idea that what Mendel has been referring was the chromosomes, right? Because there is a parallel behavior of what he called as factor and the chromosomes, right? So they have just proposed this particular idea that the inheritance unit would be the chromosomes, right? But then the experimental verification was also required. Just the proposition is not going to be accepted by the scientific fraternity, and that is the reason why the experimental verification was done by Morgan, right? So Morgan has used Drosophila melanogaster as the experimental organism, and that's how he proposed by, you know, having this particular chromosomal theory of inheritance into the real picture, that the behavior is actually parallel to what the Mendel has said, that they are being occurring in pairs, you know, perform the segregation while the gamete formation, independently assorting with each other. So those will be done. So the experimental verification was provided by Morgan. Now coming to Bateson, we could rule out this one. Why? Because when we talk about the contribution of Bateson, he is the one who has proposed the term gene, right, allele to us. Then we have Mendel, option number 4, Mendel. This could be entirely ruled out because yes, he is the father of classical genetics, who has given us the primary idea that how inheritance is going to work by proposing 3 laws like law of dominance, segregation and independent assortment. But till the time he was there, he was not able to have the proof, the physical proof, that what factor is all about, right? So that is why we could rule out this name as well. So the correct answer for the given question is going to be option number 1. Now moving ahead, we have another question, that is question number 113. That was a straight fact-based question. Now coming to the one which talks about the incorrect one for polygenic inheritance, right? So when we talk about polygenic inheritance, then we know that there will be a single character but going to be controlled by many genes. More than 2 genes for sure. So in this particular polygenic inheritance, one thing what we know is whatever number of alleles we are going to have, the effect of each of them will be additive, right? It is not just the single gene that is going to decipher that what the genotype is going to be, as we have seen in the classical Mendelian genetics. But over here, all the alleles are going to be collectively give the effect. How many times decarboxylation occur in TCA cycle would be 2 or 3 by taking link reaction. If you are considering only Krebs cycle by eliminating, see, you can understand it with this particular thing. When we talk about aerobic respiration, the first thing we know about it is that there will be complete digestion of glucose, right? Glucose has got 6 carbons, so each one of them has to break and could be released in the form of carbon dioxide, right? So we'll get back to this question, but prior to that, so if we talk about pyruvate, it is a 3 carbon compound. That means all these 3 carbons has to be eliminated in the form of carbon dioxide. So if pyruvate is formed, if you are considering link reaction, then acetyl CoA is being made, which is a 2 carbon compound. So one decarboxylation occurs over here, link reaction also known as oxidative decarboxylation of pyruvate. And then further, there are only 2 carbon are remaining for the Krebs cycle to eliminate it out. So 2 times decarboxylation occurs in the Krebs cycle. So if you include the link reaction, then it will be 3 carbon, right? 3 times decarboxylation will occur. If it is Krebs cycle, then 2 times it's going to occur, right? So this is how you will be learning the number of decarboxylation, right? Okay, so getting back to this particular question. So we were talking about the polygenic inheritance in which multiple genes are controlling a single character. It is unlike the pleiotropic gene in which a single gene is, affecting multiple phenotypic expressions. So over here, the effect of each allele of each of the gene would be additive. Like for example, there are certain characters which are which shows polygenic inheritance. Like we can have skin color in human. Like we can have human heights, right? It is also can be seen in plants like wheat kernel color, right? So when we talk about the skin color in humans, we can see a gradient of the colors, right? So if both are given in question, about the decarboxylation thing, then if it has been mentioned only Krebs cycle, then you won't have to consider the link reaction because it's a separate reaction, not the combined one. So if it is only Krebs cycle, you know the answer, it will be 2, right? So 2 would be considered as the answer. Is it clear? Okay, now getting back to this. So when we talk about the skin color in humans, they are being controlled. Let's suppose there could be n number of genes, but let's suppose for just inference, we are considering 3 genes here, A, B and C. These are the dominant forms of the dominant alleles of this particular genes, and these are the recessive ones, right? Now, let's suppose if we are having this sort of genotype, right? So this particular individual, as it is having all the dominant alleles, right? So because they are having all the dominant alleles, so they will be having very dark expression of it. Okay. How many antigens are present on human AB plus blood group? See, if it is AB plus blood group, that simply means it is having IA allele which is capable to express itself, IB allele capable to express itself, and then RH antigen also. See, if you are talking about with respect to only ABO blood group, plus an RH blood group, then you will be having 3 antigens. But in general, RBCs can have many antigens. With reference to AB plus blood group, it will be having 3, A antigen, B antigen, and of course the RH positive antigen. RH antigen, not positive. Positive is being given if RH antigen is being present on the surface. Okay. Now coming back to this particular one, so because there are dominant alleles over here, so their expression will be magnified, and this particular individual is going to have the darkest color. On the contrary, if we will be having all the recessive alleles, right, like this, small a, small b, small b, small c, as all of them are recessive, again their effect is not going to be that magnanimous, but of course as the effect of each allele is additive, but with reference to that, this particular genotype will have the lightest color. And to have the intermediate one, we will be having this sort of genotype, right? This will be having the exact intermediate between these 2 extremes. So in the polygenic inheritance, what we see is the gradients of it, right? Entire gradient of it, and depending upon the genotype, we are going to decipher that what kind of phenotype it is going to express, right? Now one more thing, when we talk about the polygenic inheritance, it's not only the genetic factor that influences the entire phenotype. Over here, the effect of environment is also really paramount. Like you could see clearly in case of skin colors, if you are living in the countries which are equatorial, right, entirely equatorial, on those countries, we'll be seeing the individuals with darker skin colors. On the contrary, the people who are living most closer to the polar countries like Nordic area, there you will see the lighter skin tone, right? So the effect of environment is also being taken into account when we talk about the polygenic inheritance, right? Getting back to this, it is saying that we have to select the incorrect one with reference to polygenic inheritance. So the first one is saying it is controlled by multiple genes, right? As the name itself suggests, poly means multiple, genic means genes. So multiple genes control this particular character. Then second one is saying it can be influenced by environment. So yes, it could be influenced by environment. Environment also have ample amount of impact while expressing the phenotype, right? Now third says it counts expression of each allele. As I've told you, the effect of each allele is additive over here. That is the reason why we say that it counts the expression of each allele, right, of each of the genes, of course. Now fourth one is saying in human skin color, the phenotype reflects the contribution of only dominant alleles, recessive alleles also which are going to contribute in it, right? Yes, both dominant and recessive alleles add up in their effect in phenotype, correct then ma'am. So this is how you will be having it, that both the alleles are going to have the additive effect, and along with that the another influence will be of the environment, and that's how the exact phenotype is going to be presented out, right? So this is how we'll be talking about it. So apart from human skin color, human IQ, human height, right, could be seen as the inheritance pattern to be polygenic inheritance, right? Okay, I hope this question is clear to everyone. Okay, so let's have the another question, which is talking about the drones of honeybee, right? Okay. So when we talk about the sex determination in honeybee, we know that they show haplodiploid type of sex determination, right? Now, how exactly we are going to say that what is haplodiploid type of sex determination? It is like the number of sets an organism is going to receive is going to decipher that what kind of sexuality the organism is having, right? So in haplodiploid type of sex determination, the males are haploid. So in honeybee, there will be 16 chromosomes that are being found in the males. On the contrary, if we see the females, right, then these are diploid and going to show the presence of 32 chromosomes, right? So we could clearly see that the males are having lesser chromosomes than females, right? So in this particular case, if the male has to produce the gametes, it will opt mitosis, as further reduction in ploidy is not possible. So it's going to produce the gamete having 16 chromosomes. And on the contrary, because the female is diploid, in order to maintain the chromosome number, it will be forming the gametes with the process of meiosis, right? So the ploidy will be reduced to half. Now, if the fertilization is taking place between these two gametes, right, male and female gametes, then there will be restoration of the chromosome number, that's 32. So this will lead to the formation of a female bee. But if this particular gamete undergoes parthenogenesis, that means in the absence of fertilization, the gamete is still developing into the organism, then it will be having only 16 chromosome and lead to the formation of a male bee, and the male bees are being called as drone, right? So that is the reason we can clearly see that these particular individuals, these males, do not have father, right? Because the mother gamete is itself is parthenogenetically developing, and same way they cannot produce sons, but they can have grandfathers and grandsons. right? So this is how the sex determination occurs in case of honeybee. So getting back to the question, if we see that over here the question is simply asking about the drones, that means the male bees. So when we talk about the drones of honeybee, the first one is saying that they are diploid, so that is incorrect. On the contrary, they are haploid in nature. The second one is saying are developed by unfertilized eggs, right? So this is true. They are being produced by unfertilized eggs with the process known as parthenogenesis. Third one is saying they produce gamete by meiosis. That's not possible because they are already haploid. Further reduction in ploidy is not possible, so that's why they cannot opt for meiosis. For the gamete formation, on the contrary, they'll be having mitosis for the gamete formation. Now fourth one is saying do not have father but can have sons, right? So this is also incorrect. Neither they can have father nor can have sons, right? So the correct one is going to be option number 2. Now, with reference to honeybee, in case of sex determination, certain times the question is asked about, certain times the comparative study or the similarity. Like for example, if I pose a question here that what would be the similarity that you could see if you see honeybee with grasshopper. So one thing that we are going to see commonly would be that in honeybee and in grasshopper too, males are having lesser chromosome than females, right? Over here if we see in honeybee, they are having 16 chromosome with reference to the females which are having 32 chromosomes. In case of grasshopper, right, we know that here we see XXXO type of sex determination where the females are homogametic and the males are heterogametic, being in term that they lack one X chromosome, so half of the sperms are not receiving it. So they'll be having one less chromosome than females. So this could be taken as a similarity between grasshopper and honeybee because in both of them males are having lesser chromosomes than females, right? So these kind of contrast things you should also notice while you are studying the chapter and specifically this particular topic of sex determination, right? If you'll ask the otherwise question that where the females are having lesser chromosomes than males, then you could see the example of butterfly where we can see ZZ and ZO type of sex determination. So females are having this time lesser chromosomes than males, right? So this is how you'll be solving the questions related to sex determination. I hope this is clear to everyone. Okay. Let's quickly move ahead to another question, and this question talks about read the following statement and select the option with the correct set of statements, and over here what we are seeing is the sex determination. So this is a very important topic in terms of questions also because multiple questions have already been appeared in the previous years. So let's talk about the sex determination thing. So when we talk about sex determination in organisms, we can decide it on the basis of chromosomes, right? If we talk about that what type of chromosomes we are having, right? Type, like for example, we can have male heterogamety, in which again we can have two types. We can have XXXO, in which males are XO and the females are XX, homogametic, right? This could be seen in case of grasshopper, as I have told you, right? Then we can have another type in male heterogamety, that is XXXY type. Here males are XY, again heterogametic, but having same number of chromosomes as a female. And this could be seen in case of humans, right? Then we can have the case of female heterogamety as well. Where we can have ZZZO type, which could be seen in the females are ZO, heterogametic, but the males are homogametic. This could be seen in case of butterfly. Then we can have ZZ, ZW types, where the females are heterogametic again, males are homogametic. This could be seen in case of birds, right? Then we could also decipher it based on the number of sets of chromosomes. So over here we could see the case of haplodiploid method of sex determination. This could be seen in case of honeybees, as we have seen, where the males are going to be haploid, having 16 chromosomes, and the females are going to be diploid, having 32 chromosomes. Apart from this, we could also decipher it with terms of temperature or the environmental conditions, we could say, right? But we have to focus majorly on the chromosomal basis of sex determination, because that's what is more important and being emphasized in NCERT as well, right? So this is how we will be having the analysis of the sex determination. Now with this information, let's get back to the question. And the question talks about that in birds, both the sexes possess 2 sex chromosomes, right? So if we come here, so in case of birds, if we see this particular organism, the males, they are homogametic but have 2 copies of Z chromosome. Same goes for the females. Over here it is, they are heterogametic, having 2 copies, but one of the Z chromosome, another of W chromosome. But the point is that both male and female have same number of chromosomes. Also both of them possess 2 sex chromosomes, whether it is the same copy or different one. Sorry. So this is the correct one, that in birds both the sexes possess 2 sex chromosomes, right? Now the B statement saying in Drosophila, females possess 2 similar type of sex chromosome, right? So when we talk about the sex determination that is being exhibited by Drosophila, that is XX and XY, same as humans, right? So over here the females are homogametic and the males are heterogametic. So because the females are homogametic, the type of gametes they are going to possess will be having the same chromosomes, right? And that's what the question is saying, that the female possess 2 similar type of sex chromosomes, that is chromosome X. So this is a correct statement. Now C says moth shows male heterogamety, right? So that is incorrect. A moth doesn't show male heterogamety. On the contrary, it shows female heterogamety. Right? Butterfly and moth both shows this ZZZO type of sex determination, in which the females are heterogametic, right? So this particular statement is incorrect. Now D statement says in butterflies, sex of progeny is determined by females, right? So always remember that whichever parent is going to be heterogametic is the one who is going to decide the sex of the child. Like in case of male heterogamety, because male provide 2 types of gametes which will be having difference, females are going to have the similar sort of genetic makeup. So that is the reason why if this X will fertilize, the one gamete that's having the X chromosome will fertilize, it will yield a female organism or offspring, while if the sperm that's lacking the X chromosome fertilize the egg, would be producing the male offspring. So in case of male heterogamety, the male will decide the sex of the individual, while in female heterogamety, as females are heterogametic, produce 2 different types of gametes, they are going to decide that what kind of individual or what kind of offspring is going to be produced, right? Because males are going to have same sort of sperms over here. So as we could see here that in butterfly, female heterogamety is present, female parent is heterogametic, and that is the reason why the sex of the progeny is going to be determined by females, right? So over here, as we have to identify the correct set of statements, those statements are being present in statement A, statement B and statement D. So option 3 is the correct answer over here, which says that A, B and D statements are correct, right? All right, now let's move ahead to another question which talks about that which of the following is not true about primary treatment of water, right? So when we talk about the sewage treatment So this particular treatment is done in 3 parts. Firstly, the physical treatment, also known as the primary treatment. The steps that we perform over here would be sequential filtration, and so there will be sequential filtration and sedimentation, right? With these, what we could remove are the particles like grit, the pebbles, and all those physical entities that could be easily removed would be done with the physical treatment of wastewater. Firstly, we'll remove that particular part. Then we undergo. So from here, of course, we'll be getting 2 things when the primary treatment is being done. One will be the sediment, the solid particles, which could be used for leveling of the ground and so many other purposes. And another thing that we are going to get from here is the effluent, the primary effluent. This primary effluent will undergo the next set of treatment, which is secondary treatment, also known as biological treatment. Now, why we say that it is a biological treatment? This is the most effective step or the treatment that we have for the wastewater, because this is capable to digest the organic impurities that are being present, right, in the water, that could not be removed by the physical processes like filtration or sedimentation. So over here we are going to use the heterotrophic microbes. This is really important. Whatever organisms are being used during sewage treatment, all are heterotrophic organisms, not the autotrophic ones. So here, firstly, when the effluent is coming over here, it's going to be there in the aeration tank where the flocks are going to be there, and there the digestion is going to occur, because these flocks are basically the aerobic bacteria along with the fungal filaments, right? This is going to form the flock, and these flocks are capable to digest the organic impurities that are being present, right? So that is being important. So you could see it will be more like, if this is, let's say, the primary treatment tank. So from here, the effluent that is going to be there is going to be passed to this aeration tank. Now the major task of this aeration tank, continuous stirring is going to be there so that the supply of oxygen will retain there, else the aerobic bacteria or the flocks that are the entire aerobic composition of the microbes, that is going to die out. So over here in the aeration tank, the main task is to reduce the BOD level of water. BOD level means, when we talk about BOD, it simply means biochemical oxygen demand. This is the amount of oxygen that is being required by the microbes to digest the organic matter that is being present in 1 liter of water, complete digestion of it. So it is directly proportional to the polluting potential of water, right? So we could simply infer the fact that more the BOD of the water, more is the polluting potential of the water, right? So when we reduce it, we are making it more cleaner, right? So this is the main task of aeration tank, to reduce BOD to the extreme levels as much as possible, right? So when this particular BOD is being reduced, then after this, this particular, the entire, output of this will be fed to the settling tank first, right? Settling tank will be there so that whatever, aerobic microbes and all other debris is going to be present, that's going to sediment out, and the effluent, which is now having very reduced levels of BOD, that can't be completely eliminated, of course, but the maximum reduction has been already achieved. So that, you know, could be gone for the water bodies. Ideally, tertiary treatment should also be performed, but this is having very reduced level. So whatever effluent is coming out of it, that is going to be gone for the tertiary treatment, right? So what is tertiary treatment? This is the third form, tertiary treatment, which is also known as physicochemical treatment. Right, in which UV rays, the chlorine treatment and all those things are being given so that it will become, you know, drinkable. It could be usable for the drinking purpose. And then finally, whatever, you know, sorry, I think I have written it somewhere else or something. Okay, I have written it in the wrong question. But anyhow, so if this particular one gone through, you know, for tertiary treatment, after this settling tank, it will go to anaerobic digester, right? And from here, some of it will go back to the aeration tank, the sediment that is being there, and majority of it will go to anaerobic digester so that those particular things could be digested well and can produce biogas or the fertilizers for the rest of the purposes, right? So if we talk about the primary treatment of water, that's only the physical treatment of water. So the only thing that is being true about it would be sequential filtration, the sedimentation and the grit removal, which is being achieved by these. Anaerobic digestion occurs during the secondary treatment of it. So that is the reason why this belongs to secondary treatment, not the physical treatment. So the correct answer for the given question is going to be option number 3. So I'm really sorry for explaining it on the other question. So that's all for this particular session. Hope you understood this, all these questions. So keep focused and keep practicing so that you can get good results. Thank you so much.